Welcome back to 6th grade lesson 3 of unit 1, part 2 notes. So this second half of notes, we're going to actually keep moving forward with how to design an experiment. The nice thing about the next step where you are looking at variables is that you've actually already identified these in your hypothesis, assuming that you were very specific in your answer choices. So variables are these factors that can change during the experiment. What you typically want to happen are only have two variables that change. One that you are in control of, called the independent variable, and one that you are not in control of, called the dependent. Again, assuming you did your hypothesis correctly by being very specific in your answer choices, then you have actually already identified both of these in your experiment. So to give you an example, we'll go backwards here for a second. In my example down here, if I use Fuji apples, then they will have more seeds. I'm in control of which apples I use. What I do not have control of is whether or not they will have more seeds. So this front half of your hypothesis is your independent variable, and this back half is your dependent variable. Now all of this lends its hand to one important thing, which is that your experiment is controlled. And we want our experiment to be controlled because if it's not, and we're changing lots of different variables and other things are shifted and not kept the same, then we will get to the end of our experiment and have no idea if it actually worked or not. So what we're going to do in class is kind of similar to what we did in the first part. The example you see on the slide in front of you, we will do together as an example. And then you will go back to the booklet that you did, and the last page will be done alone. But before you get to that alone page, you are going to do some slides on Google Classroom with a partner where you guys practice looking at different experiments and identifying those variables. The next time we were in class, we looked at step 3's continuation, which is writing a good procedure. Basically, for writing a good procedure, you want to make sure that number one, you have a very complete materials list like you see over here on the left side. And then on the right side, you will see our listed step-by-step procedure. It either needs to be bullet pointed or numbered, which way you do that is your choice. And again, when you're writing this, similar to the hypothesis, you want to be very, very specific in your wording, in your amounts, even down to, the times or how many times a step should be repeated, because the goal is that somebody else can reproduce your experiment with little to no questions about what to do. So to practice this, you and a partner are going to receive a bag of Legos. You're going to build a structure, and then on the paper provided, you're going to write step by step how you built that structure, and then you're going to take a picture of it. Once we come back from brain break, you'll trade with another group. That group will then try to duplicate your building, your structure, while you try to duplicate theirs. And then you'll each take pictures. And then if we run out of time, then the next class, the whole group, both groups will get together and make one big group. And you guys will talk about pros and cons of each other's procedures and how things turned out. Step 4, we will be spending a lot of time on this in our next unit, which is about measuring. Step 4 is where you just collect your data in a data table. That data can be both qualitative and or quantitative. And then you take that data and turn it into a nice visual picture, also known as a graph. Typically in science, we tend to stick to bar and line graphs. Again, more details and information about this particular step in our next unit. The last two steps, drawing conclusions, step 5, this is just where you kind of explain what you learned. Does your data support your hypothesis? Why or why not? And can that hypothesis or can that procedure be repeated and somebody else get the same results? This is super important, especially if you think big picture. We think about all the research that's done with cancers and different types of diseases. We want those scientists that are doing their research and experimenting to do their experiment over and over and over again, with hopefully the same results occurring over and over each time. If they're getting different results each time, that could be a number of different things that have gone wrong. They could be a faulty experiment, so maybe there's a step somewhere that was not done correctly, or it just could be that if you go back to the independent variable, whatever they're testing may not actually be the right choice. And then step 6 is just communicating, sharing those results. Obviously, if someone develops, a cure for cancer or a specific type of cancer, we would absolutely love for them to share and communicate with us, obviously, after they've gone through the repeated process from step 5. So at this point, that concludes all of our notes for lesson 3. As always, if you guys have any questions, let me know.